From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 24th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 114th episode of Matters Microbial. I am so very grateful for every listener, viewer, guest, and supporter. Hopefully, I'll continue to the next podcast milestone, sharing stories of microbiology and microbiologists each week. Please spread the good microbial word about this podcast. More viewers and listeners are always appreciated. 2 weeks ago, we had Dr. Petra Levin on the podcast, and at one point she shared her lab rules. Since that podcast, she added new ones, as seen here in this photograph. They include: be kind, do the right controls, very emphatically, look through the microscope, select growth medium carefully, and always plot bacterial growth on a log scale. I wanted to be sure to show everyone that. In other news, we're getting closer to my favorite holiday, Halloween. As you can see here, I always have microbial bioluminescent fun. I also do my best to promote good microbial PR. So when my wife, Dr. Jennifer Quinn, and I had fall break in Las Vegas, we went to the wonderfully strange Lucy Darling and Sarah Hester Ross shows. As you can see, I promoted a microbial point of view. Sarah Hester Ross received a plush bacteriophage, while Lucy has her own gigantic syphilis plush. They laughed so much, and so did we. Remember that this is the last microbiology course I will be teaching here at the University of Puget Sound before retirement. Here is the 2025 logo for this class by Liza Vandeart, and here is my last crew of classroom micronauts in t-shirts promoting that microbiology is fundamental. As mentioned in the previous weeks, how microbes respond to nutrient levels fascinates me. Here is a bacillus strain on full-strength LB. Please note the solid and distinct bordered colonies. On 1/10th strength LB, the colonies look different, slightly more diffuse. And on 1/100th strength LB, seen here, the colonies are spread out and almost feathery. Fewer cells? More surface area to take up nutrients? My micronauts want to know, and so do I. Honestly, this is what I would add to Dr. Levin's list. Just look at the colonies. Maybe just look is the right thing to say. Now, there are different populations of microbes existing on and within us all of the time. They've co-evolved with us. In addition, the members of each population interact with one another in various ways. This is the basis of the concept of sociomicrobiology, as we've discussed before. So in a way, we are all a walking, talking collection of diverse ecological niches, each with their own interactive and distinct microbial communities. Now here is a micrograph of the kind of microbes to be found, not to be personal, but on the human tongue. I'm going to be asking about this in a moment. It's a beautiful photograph. Which leads us to today's podcast guest. I'm very pleased to welcome Dr. Jessica Mark Welch of the ADA Forsyth Institute. After all, on her website, she describes oral microbial communities as, quote, bacterial high-rises, little microbial apartment buildings that these bacteria build on your tongue. Amazing. Welcome to the Quality Quorum, Jessica. It's a pleasure to meet you.
Thank you. It's a real pleasure to be here.
Oh, I'm going to have so much fun today. Before we start, would you mind telling the listeners and viewers what the Forsyth Institute is?
Sure. The ADA Forsyth Institute is a research institute. It's located in Somerville, Massachusetts, just outside Boston, and it has for really decades focused on the oral microbiome and other aspects of oral microbiology, and also now how oral microbiology and oral health impacts overall health.
It's interesting because I've had a big interest in this because the most obvious biofilm that students can study is the one on their teeth. And I make jokes about not jokes, we actually study it a little bit, about the microbes that live in their reusable water bottles. I'll have words for you about that later. But I think it's important to remember that there's a whole population, not just on the teeth, but everywhere else.
I love the way you put that we are a walking and talking collection of bacterial habitats. Yeah.
We are. I mean, I talk about it from the surface of our skin to deep inside us. It's really pretty amazing. So we're never really alone. That's the take-home lesson to me.
You have trillions of fellow travelers on your walk through life. Yes.
Exactly. I was once accused of calling students petri dishes with shoes, and that's only a tiny, tiny bit of it, isn't it?
Absolutely, for sure.
Yeah. So that first photograph that I showed, I had a couple questions about that, and it'll be up there. The YouTube viewers can see this. So can I ask first how you take that sample?
Absolutely. So we take the samples from the tongue using a ridged plastic tongue scraper, just a little piece of plastic. We just scrape it over the tongue. It's exactly the same way that you would clean your tongue if that's something that you do day to day. And then so we take the plastic tongue scraper, we scrape against the tongue. We look at the scraper and are alarmed at the enormous quantity of material that came off on it. We take that material, we can fix it in a tube. We put a fixative of some kind, ethanol or formaldehyde, and then we just drop the material on a slide, and then we can look at it with microscopy.
You're
and it's amazing, as you can see here, it's amazing what you see.
And I wanted to say, in addition, and we've done this in prior episodes of this podcast, it's possible to make taxa-specific fluorescent labels, for lack of a better term. And in the key to this particular image that I put up, they start mentioning the types of members that we see on the tongue. Would you mind explaining that just a little bit?
Absolutely. So this is what I do. This is kind of my business, is to design fluorescent probes that will light up different kinds of bacteria with different colors. So the probes themselves are short pieces of DNA. It's about 18 to 22 or 23 nucleotides of DNA. And then they're synthesized with a fluorescent molecule attached. And those probes, I designed them to target the ribosomal RNA inside the bacteria. And the wonderful thing about the ribosomal RNA in the bacteria is that there are parts of it that are conserved in almost all bacteria. So you can make a probe that hits that conserved piece. It's going to light up all the bacteria so you can see who's there, or at least who's reacting with your probe, who's accessible to the probe and reacting. And then there are parts of the ribosomal RNA that evolve more quickly. So they are more specific to different genera or even different species of bacteria. So we can design these probe sets that both have broad probes so you can see kind of the overall picture, and then we can target specific bacteria. What I do usually, so far, we've mostly worked at the genus or the family level. So we have probes targeting these somewhat larger groups of bacteria, and more now we're working on getting species level resolution so we can tell exactly who's where in these amazing biofilms.
So I don't mean to get personal about myself, but I'm so freaking old that I remember when these things were first developed, I couldn't believe the power of them. And now they're done so often, you can really answer the question, who goes there? And I really enjoy that. I do a little bit of this business with the 16S barcoding, and it's not really a barcode. I'm so delighted that you mentioned that some areas of the sequence are highly conserved versus some that are more variable. So you can look at things that are closely or more distantly related very easily. And when we look at water bottles, sometimes we pull remarkably disturbing things out of water bottles.
I spent some time trying to look at the indoor environment, the microbiology of the indoor environment, and the very best place to find microbes was, on the inside. So that little drop of water that's left in the bottom of the water bottle. Wonderful place.
100%.
They're oral microbes, by the way, of course.
Many of them, yes. And that's what I'm going to be looking for, because now I have a data set from my entire class. And now, again, we're not doing the high throughput. These are things that are cultivable on a type of medium that you understand, R2A medium. And and we get all kinds of interesting colors and all the rest. And now people are starting to bring it all together and what we're finding and why. But the power of what you do is you don't have to cultivate anything.
Right, exactly. The wonderful thing about using microscopy and targeting the ribosomal RNA, we can find out what kinds of bacteria, what kinds of ribosomal RNA are present in a sample just by grinding it up and extracting the DNA. When I first heard about this strategy, I heard about it in the context of trying to find out what was in soil. So the soil bacterial communities are enormously complicated. And so people would take soil and extract DNA. And I just thought that was the most hilarious thing. Like, what? You're going to get the DNA of the soil? Soil doesn't have DNA. Right. But there's a lot of DNA in there because there's a lot of bacteria there. And even if you can't grow them, which in the soil, for the most part, you can't, you can still, using polymerase chain reaction, you can amplify up that little piece of the piece of the ribosomal RNA that's between two conserved regions. Put your probes in there, or put your primers in those conserved regions, amplify what's in between, and you find this enormous microbial diversity, even if you can't cultivate it. So we're building on that to do the imaging. And so we can use that little, what we know about the ribosomal RNAs that are in there to design probes, even if we can't grow the bacteria. And then I should mention the reason that there are so many colors in these beautiful images is because we're going one step farther. So there are a lot of people who use this fluorescence in situ hybridization. We call it FISH, fluorescence in situ hybridization. A lot of people have been using it for decades, using usually 2 or maybe 3 probes and looking at them with a microscope that's got red filters, green filters, blue filters. So you can basically see 3 colors. And the extra step that we've taken to produce these images is we're using a fancy, expensive microscope that can read the spectrum from every pixel in the image. So because it can read a spectrum, we can use these different fluorophores that have overlapping spectra. So we have 3 different ones, and they're all different shades of red, basically. And there's some others that are different shades of green. But by reading the spectrum, you can tell them apart, distinguish which is which in the pixels of the image. And that way we can use 8 or 10 colors pretty routinely. So we get to label most of the major bacteria in any sample, and you can really see how they interact with each other, who's next to who, what's going on.
So in a way, it's biogeography.
It's absolutely biogeography, just at a really small scale, microbiogeography.
And what I like about this so much is that when I was in school, and I get students who'll ask questions like, were you around when microscopes were invented? Yes, students, I was. Right. Anyway, we would think about looking at Streptococcus mutans on your teeth and how it reacts to sugar and all the rest. And those were always fun to see. But again, the majority of organisms that we want to study, now this is controversial, but I'm going to say it anyway. The majority of organisms in nature don't grow on laboratory media very well. Now, my friends who, and I'm going to call them the cultivarists, insist to me that they find just the right medium, they can get everything to grow. And that might be true. But as far as what most people can do easily, that's a very limited number. So if you're just looking at what you can grow, that's quite limited, and it doesn't answer all kinds of questions. For example, the juxtaposition of where microbes are. So on the tongue section that we've been discussing, you could easily come up with models just like in dental plaque where microbes are interactive with one another. And we've talked about that in other episodes, and it's fascinating. You know, there's the whole William Blake poem about to find a universe in a grain of sand. Well, this is a universe in a tongue scraping.
Yes. And it is a bit alarming what you can see and how much of a universe there is in there. And one thing I want to say about that image that we're looking at is that you can see the host tissue as well. So this is an image, it kind of looks like it might be a cross section of a tongue, but it's actually a cross section of a much smaller object. So your tongue has papillae. There are fungiform papillae that contain the taste buds. And then there are filiform papillae, and nobody talks about them. They're about 10 times more abundant than the fungiform papillae, and they have these projecting hairs. And in humans, those projecting hairs get thickly coated with bacteria. So what you're seeing in the image, the sort of gray stripe down the middle, that's the projecting hair, and then each little blob on it is a individual bacterial cell in that tiny little object. So one thing that we can see in that image is also the relationship not only of the bacteria to each other, but the relationship of the bacteria to the host, who's binding right there on that host tissue.
Well, this is the thing that people just, they'll say things to me like, and again, I am not making light of anyone who's new to microbiology, but they'll say, oh, well, they're sticky. Sure, but that's not the point. The point is we've co-evolved with them, so they have specific associations with anatomical structures. So when you read about the farmer ants, the leafcutter ants, and you discover on the ants is a specialized structure on which the Nocardia bacteria tend to colonize, I would argue the same thing's taking place on your tongue with different microbes.
Yeah, absolutely. And you really see that specificity of the interaction. So they're not just broadly sticking to anything. These bacteria have receptors that they very specifically bind to one another. And the way that you can, a reflection of that in the mouth is that you have almost entirely different sets of bacteria living on your tongue and living on your teeth. And there's other bacteria that only live on the roof of the mouth, right? There's bacteria that are very common on the cheeks, on the cheek cells. So all of these different habitats, different structures in your mouth are colonized by different sets of bacteria, even though obviously they're all falling off into saliva and moving around. There's no colonization barrier, right? They can get from one, there's no accessibility barrier. They can get from one place to another easily, and yet the bacteria on your tongue are basically entirely different from the bacteria on your teeth.
That's just amazing because, you know, we've talked about the gut microbiome and gut microbiota quite a bit on this podcast. And it's interesting to consider what you just said, different populations on the tongue versus the teeth, even though they're awash in the same ocean, as it were.
Yeah.
And you know, by the way, one of the things that I delight, and I don't talk about it, I send it to students and ask them to think about it, and then they tell me. There's a paper from perhaps 10 years ago, I'll put a link to it in the show notes, about microbes transferred through, please forgive me, intimate kissing. This is from somewhere in Northern Europe, and it's just an amazing paper. And it reminds me of an artwork that a friend of mine made where it showed two people holding hands and little dots of different colors representing microbes. So when we're holding hands, so are they. So we really are in a sea of microbes within us and without us. And what happens at the intersection is very interesting to me. And again, it comes back to microbiogeography.
Yeah, absolutely. And so, one of the interesting things we've learned about the oral microbiome in particular is that we each individual person has basically the same species of bacteria on our tongues. But we all, nonetheless, if we were to sample from, 100 different people in a room, and then a year later they all come back, we sample from them again, we could identify who's who based on their microbiome, because although the species are pretty much the same, the strains are different, right? We all have different strains. And how that gets set up is an interesting field of research. There are people who study how you gain your microbiome, from birth moving forward, and then how it's affected by what you do, who you're married to, and we did a very small study of 3 couples, and we studied their microbiomes. and with sequencing and looking at the strains. And then we gave the data for analysis to somebody. We didn't tell them who was married to who. And they figured it out, right? And, because there was a certain amount of strain sharing, not total homogenization, but enough strains get transferred from one microbiome into the other that you could identify people who associate with one another.
And we're not even discussing the interactions that those, and I'm going to call them the tongue microbiota, for lack of a better term. We're not even talking about the interactions that they can have with one another, either positively or negatively. It really is. I mean, I often tell students the gut microbiota is kind of like a rainforest. But honestly, so's the mouth. And
I would say the mouth is even more a rainforest than the gut microbiota, because the mouth is so highly structured and there's so much diversity from place to place. So we've been looking at this one amazing image of the tongue microbiota. We have another amazing image of a different object from the tongue that you can see. And what's interesting about that is it's a larger structure, and it's at the center are these filaments. We've colored them purple and yellow and green, all these different sort of long filamentous bacteria. And what we notice there is that you see these same kinds of bacteria regularly together in these structures. So this is work of Tabita Ramirez Puebla, a really wonderful postdoc who's been working with me for several years. And she discovered that when she looked at these larger objects, which she had to do by embedding them in plastic and then making thin sections and then putting this little thin section on a slide and hybridizing the probes to that, then she could identify these taxon associations. And that's really important for us to understand, because there are hundreds of kinds of bacteria in the mouth, and there's dozens that live on the tongue, and there's dozens that live on the teeth. We want to be able to bring them into the lab, grow them together, and find out how they behave differently when they're next to this bacterium versus that bacterium. But there are so many possible combinations that you could pick. Like, how are you going to know where to start? And the best way to know where to start is to look at what's really going on in the mouth. Which bacteria do you find next to each other? Right, those are the ones you should start with. Put them together in a test tube and see what happens.
There's some work from Joe Handelsman's group at Wisconsin-Madison about what she calls the Thor system, which is an assembly of 4 different microbes that show staggering diversity in interactions with just 4. And so I would say that this is a demonstration of the depth and breadth of the microbial world and the interactions upon them. And we tend to be reductionist by nature. We want to look at one thing by itself, but that's not how microbes live. And that's what we're discovering everywhere we look.
Absolutely.
I did want to say parenthetically that when my students swab their water bottles, sometimes they get really unfortunate looking things and other times they don't. So I would imagine that a early morning scraping of the tongue would give you a different appearance than in the middle of the day.
That's a really interesting hypothesis. And I would say that the bacteria, actually a lot of the bacteria on your body and probably the bacteria in your mouth, they do have diurnal cycles. Every time you scrape your tongue, you basically see these big, amazing structures. And in fact, somebody once tried to calculate the microbial load on the tongue by scraping and then dilution plating to try to count how many bacteria there are, and then scrape again and dilution plate and scrape again. And what they determined is that the microbial load on the tongue is infinite. Which obviously can't really be true, but you keep scraping, you keep coming off. So yeah. I would say no matter what time of day you do the sampling, you'll find a lot there.
So one of the things I make a joke of this. I have a lecture I give on what I call the heresies of microbiology, by which I mean things that are true about microbiology that many people don't think about other than microbiologists. Mm-hmm. And it's especially important for undergraduates, which I teach, to know about that. So this idea that bacteria grow in pure culture in a 2059 culture tube is wrong on both levels. Mm-hmm. And so
they evolved in communities. They evolved to live in communities, and that's how they expect to be.
And generally on surfaces. And, and so that's what we're talking about. So this issue of surfaces is important. And I want to say many times I kind of nerd out over really interesting microbial questions. I call that WTM for what the microbe, or OMG for overwhelming microbial greatness. And all of that's true. But this issue of adhesion and developing a surface structure is relevant to issues of health as well. So this is not just of academic interest, it's of health-related interest as well.
Yeah, absolutely. And one of the things, well, so maybe first I'll say that I think that we co-evolved with these bacteria. And so and then the human host, modulate the bacterial community in many different ways. It involves our immune system, antimicrobial peptides, but it also involves feeding them with saliva, right? Giving them the kinds of molecules in saliva that are going to foster the growth of the bacteria you want and discourage the growth of the bacteria that you don't want. And one of the things that we're learning about the oral microbiome, about the function of the oral microbiome in the human body, is that oral bacteria are involved in regulating your blood pressure. And the way that works is that, so if you eat a diet that's rich in green leafy vegetables and beets and celery, it turns out that's a high nitrate diet. And it lowers your blood pressure, but not if you're using an antiseptic mouthwash. Isn't that amazing? So what's happening is that the nitrate that you ingest gets converted into nitric oxide, and nitric oxide dilates blood vessels and in fact can improve athletic performance, apparently. So athletes are starting to drink beet shots right before they do a competition because that apparently improves blood flow. But the way that nitrate gets converted into nitric oxide involves first conversion to nitrite. Your body does not do that very efficiently, but there are bacteria in your mouth that can do it very, very well. So when you ingest a high nitrate meal, that passes right through your mouth pretty quickly. The nitrate gets absorbed into your bloodstream. Your body then concentrates the nitrate into saliva tenfold. So you get a salivary nitrate concentration that's tenfold the plasma nitrate concentration. And that in saliva, that nitrate washes over the bacteria. So your salivary nitrate levels go up after a high nitrate meal. They go up for 4 or 5 hours maybe, and then they come down. And during that time, the bacteria are converting nitrate to nitrite, and that modulates blood pressure. So it's a combination of diet and oral microbiota carries out this really super critical, very important part of human biology.
So I want to say this very, very cautiously. So how we would therefore use mouthwash might matter in ways we hadn't considered? Would you say that?
Yes, I would say that we should consider much more carefully whether we want to be wiping out the oral microbiota regularly with mouthwash. There are reasons. So if you have periodontal surgery, a lot of times the periodontist will then prescribe that you use an antiseptic mouthwash for a couple of weeks afterward or something while you're recovering from the surgery. Regularly using mouthwash might not be that great an idea. Although I should also say, again, saying somewhat cautiously, it depends a lot the kind of mouthwash, right? Are you actually killing the bacteria or are you just doing something else, right? And that is really an area that needs a lot more research. I would really love to study what is actually happening when you're using mouthwashes of various kinds.
I can sense the ghost of my late mother behind me, insisting I told you so, because she talked about saltwater mouthwash an awful lot with me.
Mm-hmm. Yeah. There are various mouth rinses. Yeah, the ones that actually wipe out the bacteria may be kind of a limited subset of them, and that's where you, I think, want to.
And this goes a little bit to microbial PR, because people have talked about the relationship between periodontal disease and porphyromonas in particular, and a possible association between heart issues. And that's not the topic for today, but what I am trying to say is that everyday listeners and viewers are only dealing with what the media tells them, which is why I enjoy doing this podcast a little bit.
Yeah, absolutely. I should put in a little plug for the bacteria on your teeth, because we also have gorgeous, gorgeous images that we've been able to acquire of dental plaque bacteria. And I mention this because there is a bacterium that sometimes lives in dental plaque called Cardiobacterium valvarum. So that doesn't sound very good, because it's basically said this bacteria is named basically heart valve, right? Or a heart valve bacterium. And so, okay, first I'll talk about the happy bacteria that grow on. So I was talking earlier about the selection, your body selecting for the bacteria that you want to have growing on your tongue and on your teeth. Among the bacteria that seem to be health associated are bacteria called Corynebacterium, and we've discovered by imaging that these Corynebacterium, they form these beautiful bushes on your teeth. So they root, there's kind of an ecological succession that happens on your teeth. So when you go to the dentist, they clean your teeth, and they really do get quite clean. And then immediately the teeth get coated by saliva, the salivary pellicle, glycoproteins that stick to the enamel. And then there are certain bacteria that are very good at attaching to that salivary pellicle. Streptococcus and actinomyces are among them. So they form a little biofilm. And then after about 24 hours, Corynebacterium comes in and binds. So we suspect it's probably binding to the actinomyces or something, right? But then Corynebacterium grows up and out, forms these beautiful big bush structures. And then at the tips of these big, beautiful bush structures are structures called corn cobs, where there are little cocci, little round bacteria that stick around the tip of the Corynebacterium. And some of those cocci, Streptococcus cristatus, for example, has been shown to inhibit one of the periodontal pathogens. So Porphyromonas gingivalis is a bacterium that a lot of people study because it's involved in periodontitis. It's good at eating proteins, which is kind of bad news if it's down in your gums. And Porphyromonas gingivalis has a harder time forming a biofilm if Streptococcus cristatus is around, right? So this is an example of ways that your body might be encouraging the growth of bacteria that have inhibitory properties against the bad guys. And then, okay, so back to the heart valve. But so among the bacteria that you can find in your dental plaque are bacteria that have also been found in heart valves and causing problems. And it turns out that the bacteria in your mouth, they fairly frequently get into your bloodstream because they're right there, on your gums, right next to your gums. When you brush your teeth, you get some little abrasion or something. The bacteria get into your bloodstream. Normally, they're just cleared. The body has mechanisms to clear them out of your bloodstream to try to keep your blood sterile. But the bacteria can end up lodging in other parts of your body and cause problems. When we apply these same methods, these same kinds of probes to light up the bacteria that we see on the teeth in dental plaque. So we collect these samples just by using toothpicks or floss, or we get professional clinicians to use their tools to scrape plaque off the surface of the teeth. We fix it in a tube, drop it on a slide, apply probes. And boy, we look through the microscope and we see these amazing structures. You can see two of them here. So we call these hedgehog structures because of the kind of spiny nature of these filaments. So the bacteria that you see here, again, every color is a different kind of bacterium, and we false colored the bacteria. So the images we get are black and white of each bacterial type. We false color them and overlay them so you can see the relationship of the bacteria to each other. And what we find is that Corynebacterium, which we have shown here in magenta, it makes these bushes, these really dense bushes that really create a spatial structure. So in a way, looking at these images tells us that the Corynebacterium, it's kind of like the oak tree in the forest or the coral on a coral reef, right? It's creating a spatial structure that other bacteria inhabit. So in the picture on the top, you see around this bush of Corynebacterium is a ring of mostly green bacteria. Those are streptococci. And so they're living at the surface of the plaque. The streptococci don't mind if there's oxygen around. So they create this little layer at the surface. They use up the oxygen. And then on the interior, you see in sort of light blue and yellow, those are Leptotrichia and Fusobacterium, two different kinds of bacteria. They like a mostly anaerobic environment, and they find it under the surface in the plaque because the bacteria at the surface are using up the oxygen. And then the image at the bottom, you just see two of these beautiful structures that were next to each other in plaque. And then in between, you have this little row of this little layer of streptococci forming these corn cob structures.
And it emphasizes this bit that I think everyone forgets, is that it's all about surfaces, and bacteria can be surfaces for other bacteria without necessarily having positive or negative effects. Surfaces are surfaces.
Yeah, that's absolutely right. And surfaces are, if you, a bacterium, are finding another bacterium to bind to, you might select which other bacteria to bind to based on the environment that's going to put you in, right? So bacteria might evolve to bind to this Corynebacterium bush because other bacteria also bind there, and that creates a certain environment that a bacterium is well adapted to.
It's like a wimpy person hanging out with a really strong person. Protection.
Potentially, it's exactly. Protection and creating an environment. It's like hanging out with the cool kids. This is the right place to be.
Yeah, or the mouthy kids in this case. Oh, that's bad. I remember a time when people would talk about a negative interaction between microbes and human cells. Just bear with me for a second. And then folks objected to that Marshall viewpoint. I'm not making a judgment. That's what they called it. And what I'm going to call a kind of a Margulian approach happened, where everything was happy and interactive in a positive way. And that made me uncomfortable because I've learned that life isn't very binary that way. And I would say that it's very much like an ecology. And there are different interactions, and they can change from time of the day or other environmental stressors or conditions in general. So teasing out what's taking place is very challenging. But the first part is to ask what's there. You've already mentioned that not only, and this is fascinating to me, not only are you saying that there are microbes that attach to particular structures in the human mouth, but then there are other microbes that attach to the first microbes in a very specific way. Yeah. In other words, it's microbes all the way down.
It is absolutely microbes all the way down. Yeah. And most of the surfaces in the mouth would get coated pretty quickly with microbes. So most of what the other microbes are binding to is the first microbes. Mm-hmm. And that suggests, interestingly, actually, that if we're trying to figure out how it is that you get different bacteria, more or less completely different sets of bacteria on the tongue and the teeth, a lot of it is going to depend on who binds first, right? And then, if who binds first is a pretty specific thing, then who binds to them can be specific, and you can build out these very different communities starting from that different foundation. And then there's a question, okay, so do we, the host, create that different foundation? Yeah, so I mentioned the salivary pellicle that happens on the teeth. There's also a mucosal pellicle that happens on the mucosal surfaces. I think much less is known about the mucosal pellicle, and maybe much more should be known about the mucosal pellicle because it's probably pretty critical in establishing these really important bacterial communities. One thing I want to say about whether the bacteria are cooperating or not cooperating in your mouth, we took a closer look at these corncob structures that grow at the tips of the Corynebacterium. These corncobs, again, they had been seen in the 1970s with electron microscopy, and like, oh, look, that's when they were given the name corncobs, dental plaque corncob structures. And people did some microdissection. And what they got out from microdissection, then they microdissect a little thing and they try to grow it on a plate. And what they got was Corynebacterium montruchati, that big, beautiful thing that we color magenta in our pictures. And then Streptococcus cristatus that I was just telling you about that inhibits Porphyromonas gingivalis. So people were saying, oh, okay, so corn cobs are those two taxa. When we used our new microscopy technique where we could see lots of bacteria at once, suddenly we see, there's more going on there. There's a second whole genus of bacteria, Porphyromonas, that's also binding directly to those Corynebacterium filaments. And that's a little weird because the Porphyromonas that everybody studies is Porphyromonas gingivalis or Porphyromonas endodontalis, and they're both pretty strict anaerobes. And yet you've got this other member of the species hanging out there, at the surface. There's going to be air at the surface. There's going to be oxygen at the surface. Okay, so there's Porphyromonas in addition to Streptococcus. And then there's another bacterium that gloms onto the outside. We think it's an aggregatibacter. It's either a Haemophilus or an aggregatibacter. But yeah, so it's a more complicated structure. And then you get to start asking questions like, okay, are they competing with each other for access to that filament? So you want to bind to the tip of the filament so you can hang out in the saliva and do whatever it is that you little bacterium do. Are they competing for a binding site, or might they be somehow getting something from each other? Might they be trying to recruit each other down out of the saliva onto this surface that they've got? One thing that we do know is that the streptococci, at least some of the streptococci, they are aerotolerant. That's one reason why they're hanging around on the outside of this structure, and they use up the oxygen, but they're not using it to breathe. What they're doing is taking the oxygen, turning it into hydrogen peroxide, and trying to kill their neighbors. And then their neighbors who live there regularly, they have an enzyme that detoxifies the peroxide because they know, there's always these strep around. They're always making this peroxide. You got to get rid of that in order to live here. So yeah, it's not simple.
How do you keep from calling that something akin to chemical warfare is beyond me. But I understand the other side of it, that we tend to put our human views on these associations. And to me, a lot of it is just jockeying for surface area, And
go
ahead.
It's jockeying for surface area, but it's also they need each other, right? So bacteria, they have very small genomes, especially bacteria that live in the mouth have genomes usually about 2 megabases or so. That's pretty small in the bacterial world. And the way that they get away with that is they get rid of a lot of the genes for synthesizing stuff you might need, vitamins or whatever, because they know that somebody near them is going to be leaking that thing and they can take it up, right? So that only works if, again, they've evolved to live in this community. They basically have to live in this community. And that's why some of them are so hard to grow on a plate, right? If you scoop them out on a plate, that plate doesn't have whatever it is that they expect is going to be leaking from one of their neighbors, then they're not going to grow, right? So on the one hand, they're jockeying for space, but on the other hand, they need each other.
It's really interesting because the saying in real estate is it's location, location, location. And I would honestly say it's true here, but I would call it context, context, context.
Mm-hmm. Yeah.
And we are so wedded to looking at one thing at a time. And I understand the value of doing that initially, but that's not how the organisms normally live. In the early days of biofilm studies, I would, when I was reviewing papers, I would say, well, this is just, let's just say, like I used to do, they're making biofilms with Pseudomonas putida, and they're nice and reproducible, great data sets. And then a criticism I would make on my own work is that's just one organism. And I would say that in reviews, and then the response would be, well, like in current experiments, we're looking at 3. And then I'm thinking, but that's, again, not how they live. But you have to start somewhere.
Right. You have to start somewhere. And it's even when you go from 1 to 2, it gets so much more complicated. And then you add 3 and 4, and the complication just goes through the roof.
Yeah, just like Joe Handelman's work. And as you said at the beginning, soil microbiology is so utterly complicated, and it makes sense that it is, just like what we're talking about with, say, the human gut or the human mouth. But it's not the way most people think. And that's the value of what you're doing here in microbiogeography.
Absolutely. And this is a good time to mention that these techniques that we use to look at bacteria in the mouth, we can also use them to look at bacteria in other environments. I'm always on the lookout for cool samples that have bacteria doing interesting things. So one of the places that we've looked is in the gut. And we've done that with a model system, so a previously germ-free mouse into which we introduce bacteria characteristic of the human gut. This was work that we did with Jeff Gordon at Washington University, St. Louis. And there, what I thought was really interesting about that is that the bacteria in the gut were much more mixed. They had a much lesser degree of sort of obvious spatial organization. It looked much more like just a bioreactor. So I've started to think about the gut more as sort of a partially mixed microbial bioreactor. So in our image from the gut, you can see this is a mouse gut, the gut of a previously germ-free mouse that was then inoculated with bacteria characteristic of the human gut. And we've labeled the different kinds of bacteria with different colors. So you see lots of little colored dots here. Each of those different colored dots is a different kind of bacterium. And then there are these yellow blobs. Those are pieces of food that the mouse was eating, right? So in this image, you see in the lower right, there's host tissue, a sort of big gray thing. That's the epithelium, the gut epithelium of the mouse. Then there's sort of a gap where there would be mucus. I haven't stained the mucus that's going to separate that host epithelium from all the bacteria in the center. And then you see these globs and swirls of bacteria. And one thing that you can notice is that there are different patches of colors. So some of the bacteria, the ones that are labeled blue and green and purple, they seem to like to live right in and right close to that mucus layer, pretty close to the host tissue. And then a little bit farther out, you see more of the bacteria that we've labeled in red and orange. And they seem to be a little more abundant near the food. But then you also see patches of the blue bacteria toward the center of the gut. And I think that's a reflection of the fact that you're getting all this mixing. So all the material in the middle of the gut is getting pushed up against the wall of the gut, mixed with the mucus, pushed back. So really, you see the bacterial communities that thrive in mucus, you see them all through the gut. And in this picture, you see the little, here there's sort of a low magnification view, and then some insets that give you higher magnification, where you can see that each individual bacterium is really, it's next to other bacteria of a different type. So when we talk about this gut, I like to think of it as a partially mixed bioreactor. When we talk about partially mixed bioreactor, it really all those bacteria are being mixed by the host at that cell-to-cell level. I think that that's the way that we mammals have co-adapted with our gut microbiota. We don't want them forming biofilms on the inside of the gut, right? We want them to be in there digesting helping us digest our food. We're going to keep kind of mixing them around, and then we're going to push them out before they have a chance to do anything.
This is very interesting to me personally, because I've always worried, and I've done a little bit of work on microbes within lizard cloacae. Just bear with me for a second, okay? Mm-hmm. I know it sounds very personal, but what I was going to say is that I would be concerned if I just look at a fecal sample that's not representative of what's adhering to the epithelia. And if I'm understanding you correctly, I need to change my viewpoint. So I think of the gut, as you say, as a partially mixed bioreactor. So perhaps the epithelia isn't as important as we might think as an attachment site.
Mm-hmm. So in my opinion, for the human gut, yeah, the goal of the host is to stop the microbes from attaching. Now the mechanisms for that, there's the mucus, which acts as this big sticky net, and we are secreting an enormous amount of mucus into the gut, right? Constantly secreting mucus, constantly creating flow. When I was in school, I learned that the function of the colon is to absorb water. right? And that is just such the tiniest little fraction of what's actually going on in the colon, right? Especially the proximal colon.
Yes.
So you're having all this the mixing, sort of the motions that serve not only to mix the microbes with one another, but they push the microbes up against the wall of the gut where they get mixed with the mucus. And I think that's super important because that allows the mucus to kind of permeate the whole microbial community and affect microbial biology. Like, for example, microbes in the gut, when they're in the presence of mucus, they can't express their flagella, right? So they can't be swimming around independently. They're kind of kept planktonic, right? So they're okay. There's mucus that's changing the biology of the microbes, and the structure of the gut is designed to mix the mucus with the microbiota. The epithelium of the gut sheds rather rapidly. So does the epithelium in your mouth, right? Your cheek cells are shedding all the time, right? So if bacteria do manage to get to glom on to the epithelium, then it gets shed, right? So there might be microbial strategies to evade some of this, like spiral bacteria can corkscrew their way through the mucus so they don't get captured as much. So some of the, like in the mouse, there are these mouse intestinal bacteria, and they seem to be able to get through the mucus and actually get to the host wall. But most of the microbes, I think, are being kept in this planktonic state, and then in the mouse they get rid of them pretty quick. So what was so surprising to me about the lack of structure in the gut was that, you know, it's warm in there, there's plenty to eat. You would expect the microbes to be dividing, like we all know, you know, so E. coli can divide in 20 minutes, right? You know, so you would expect that just because it's so comfortable in there, the microbes would be forming little microcolonies. And yet we didn't see that. We saw these individual little bacteria, basically, which suggests that the mixing forces are faster than the ability of the microbes to divide.
It comes down to physics, doesn't it?
Yeah, physics and, yeah, it's really biophysics, most of it.
Yeah, and this idea of interacting in both directions, a tissue with the microbe with the tissue, is what I tell students is the fact, because it's a coevolutionary relationship. And you mentioned mucus. There are some organisms that require it to grow because they can actually break it down. Mm-hmm. And there are some people who think, and they've changed the name, this is what happens as I grow older, from the Bacteroides to another thing that I can't yet pronounce, but you're with me on this. Some I think it's micro. I'm going to say it incorrectly, but you know the organism I'm speaking of, the taxon. There's some evidence that it kind of tickles our epithelia to make more mucus. So in essence, they're mucus farmers, and we're the farm.
Absolutely.
I just love that.
There's some work, I think this was work out of Jeff Gordon's group in Washington University, that if you take a germ-free mouse and you introduce gut bacteria, the gut bacteria signal to the host to produce the kind of mucus that they want to eat. So yeah, definitely a two-way street. And so, okay, so interesting question. How much of that is going on in our mouths and in our gut? Yeah. Yeah, for sure. Okay, I have to show you one other cool thing we can do with this kind of imaging, which is to look at marine organisms. So I was working at the Marine Biological Laboratory until about 3 years ago, and one of the things that we did there was look at the bacteria growing on kelp. So this was in collaboration with Catherine Pfister at the University of Chicago and her student Brooke Weigel at the time. So they collected kelp and just fixed it in some ethanol. They just sort of took these pieces of kelp and sent them to us. And we used our probes to, again, we first embedded this kelp in plastic and made cross sections, and then used probes to look at the distribution of bacteria on the kelp. And there too, you find interesting spatial structure. You can see some of the bacteria sort of projecting from the surface into where the seawater would be, and then other bacteria that are kind of digging down into the kelp, different kinds of bacteria doing different things. So it's just another example of how looking, actually seeing what's going on, can give you clues about what these bacteria are doing. We've learned so much from sequencing, and the first thing you do when you sequence, you take a sample, you grind it up. You destroy the spatial structure. And if you preserve that structure, it's really amazing what you can learn.
So I'm going to try and pressure you to look in a water bottle straw.
Yeah.
if you're ever going to do this kind of stuff on weird samples, there you go, because I'm just doing stuff that I can grow. And I don't have the funds to do any deep sequencing. And even if I did, it would tell me nothing about the microbiogeography.
Mm-hmm. Yeah, it's probably oral bacteria, and who knows what they're finding to grow on in your straw. Yeah.
Yeah, and actually, I would go farther. I will bet you, and I will say it right here, right now. I will bet you $100 that if you do this, you're gonna see little pits on the plastic where some of these microbes are breaking down the plastic, because I see methylotrophs all the time in there. And I would not be at all surprised. And that's fascinating to me. I don't mean to nerd out, but it's how I feel.
Oh, yeah. Bacteria are just amazing.
Yeah. A few weeks ago on this podcast, we had a guest who was discussing that microbes could be recovered from the dental calculus of, I'm going to say, historically ancient humans as well as Neanderthals. Do you think it would be possible that we could study other aspects of the oral microbiome in a similar fashion?
That's a really interesting question. And it's amazing what they're able to do to extract bacterial DNA from this dental calculus. I think it would be harder to preserve the bacteria from the tongue or from the cheeks. I think the dental calculus is special in that the bacteria start to mineralize even in life, right? So I think that preservation is something you're only going to find probably in plaque, but it's really interesting to think about using that kind of information to find out how the oral microbiome has changed over time, and all this toothbrushing that we're doing, how does that change it? And I think what they're finding is that it tends to sort of shift the microbiome to keep the microbiome in a more immature state.
Huh.
Is that good or bad? We don't know.
It just is.
Yeah.
So is there evidence related? Is there evidence of, I'm going to just call them tongue microbes, please forgive me, tongue microbes into the dental calculus?
That's a really good question. We don't see 100% separation. So even in samples from modern people, samples you could take today, the reason I say that there's a almost complete separation between tongue and plaque bacteria is because some bacteria are 100-fold more abundant on your teeth than your tongue, and there's others that are 100-fold more abundant on your tongue than your teeth, and we figure there's a little bit of transfer. So you're going to find a few bacteria from tongue and plaque and the other way around. So yeah, actually you could find out a little bit about other parts of the mouth from what's stuck in the calculus. That would be really interesting.
And it's so funny because you use the word flow repeatedly, and it's flow. Same thing as the gut, but more, I think, more distinct microbiogeography, in my opinion. Yeah.
And flow is one of the most important concepts because it's one of the things your body can control that then controls the microbiota, right? The flow of saliva means that all the bacteria who want to live, to anthropomorphize them, if they want to live in plaque or on your tongue, they have to hang on, because otherwise they're going to end up in the stomach, right? And they don't want to be in the stomach. So all the bacteria, that's one reason we have so much really cool spatial structure in the mouth, is because all the bacteria are stuck to each other. So they evolved to stay stuck. And the rate of flow through the gut is one of the things that's going to control how much the bacteria get to stick around in the gut, how long they're in there, how much time they have to do their thing.
With ample respect to all the people who write microbiology textbooks, and notice that I do not, they're often, this is not given enough interest in the role of attachment other than in disease state. And I think that this is a really critical idea. So I'm glad to hear you repeat it.
Yeah.
Speaking of mouthwashes, is it possible for some of the tongue microbes, please forgive me again, that isn't specific enough, but you know what I mean, to become resistant to some of the antimicrobial components that are there, and then in turn, through horizontal gene transfer, move that to other microbes in the mouth and maybe on the way down?
Yeah, that's a really interesting question. And I guess it would depend what exactly is in the mouthwash. So there's certain things that microbes can become resistant to, and other substances that just wipe them out rather broadly. What I do know is that antimicrobial resistance genes are handed around from bacterium to bacterium in the mouth. And in fact, a biofilm in general, is a great place to hand around antimicrobial resistance genes. Bacteria break open, they lyse, they spew their DNA everywhere. Other bacteria take up that DNA, partly just to eat it, to take it apart and use its components, but also they could try to recombine it into their own genome. So biofilms in general are a great place for that to happen, and the mouth is among them. And there are plenty of antimicrobial resistance genes in the mouth. So it would, certainly anything you do to perturb the community could have downstream effects. But I should also say those antimicrobial resistance genes, they find them in the Neanderthal plaque. So they've been around. We did not cause these genes to occur. We might be causing them to become more frequent.
I think, yeah, and this is a level of narcissism that we have because, again, first evolved, last extinct. We have pressures on the microbial communities, yes, but they were there first. That's the important thing. So it sounded to me a little bit like there's a balance between the necessity of keeping the teeth clean versus the tongue microbiota. And is it a trade-off, or am I misunderstanding it?
Yeah, I don't think it's a trade-off because the bacteria are really good at coming back really quickly. So you can brush your teeth. You can and should brush and floss your teeth. Keep your teeth clean. Your tongue is going to be pretty much completely unbothered by that, right? And then you can scrape your tongue, and that scrapes off presumably the kind of older, more developed clusters of bacteria, but new clusters come right up behind them. I'd say so. Even when you use an antiseptic mouthwash and wipe out the bacteria, you wipe out the surface layer of bacteria, but not the ones that are down under, with an antiseptic. Maybe in a few hours, they're back. So don't worry about them. You can mechanically remove them all you want. They're going to be fine. That's a great question. Thank you.
Thank you. So I'm watching our time pretty carefully. So are there more things? Because I got a little carried away, but I hope that's all right with listeners and viewers. And you and I had a good time anyway. Do you have any other things about your research program you want listeners and viewers to hear about?
I think we covered we covered the key points.
Perfect. So let me ask another question. Mm-hmm. Would you care to share your microbial journey?
My microbial journey. Yeah. I came late to microbiology. So when I was in college, I studied evolution. I was really interested in evolution. I did my senior project in paleontology, and it was great. It was amazing. And in graduate school, I was interested in the evolution of genetic systems, trying to figure out actually why animals have recombination. Animals, generally, there's males and females. Why is that? Why don't we just clone ourselves? So we were studying, what is in fact the purpose of recombination. So I was just minding my own business doing this basic biology. But when I went to the marine biological lab, everybody around me was studying bacterial communities. They were finding this incredible diversity. It was just when the sequencing was becoming cheap enough that they were developing methods to look at the incredible diversity of bacterial systems. And then there were people over in the ecosystem center studying how bacteria really run the planet. I mean, so here I am trying to study animal genetics, and everyone around me is doing this incredible work with bacteria, and I just got sucked in.
It's the one true microbial faith. That's how it is. First evolved, last extinct. Am I wrong? I am not.
No, you are not wrong. Yeah, I have the enthusiasm of the convert.
That's why I want to call myself, for my retirement job, a microbial evangelist.
Yeah, because microbes really do. Microbes run the world. It's
so
really amazing.
That's a misstatement of the Beyonce song, "Who Run the World," and we all know it's germs, right? There you go.
There you go.
That's a wonderful story. So as far as what we've learned today, we've learned that there's incredible, dizzying complexity of microbes within the mouth. And that isn't simply in terms of number or types, but in terms of spatial organization. And so do you have any advice for people as they look in the mirror every morning with a toothbrush in hand, or Lord knows a tongue scraper?
Yeah. And actually, I've got to tell you about the microbes of the mouth. It's actually one of the simpler bacterial ecosystems out there. One of the reasons we love to study it is because there's, only 2 or 3 dozen taxa that dominate the mouth, right? So yeah, or at least dominate the 2 or 3 dozen dominate the tongue, and another 2 or 3, 3 or 4 dozen dominate the plaque, and that's pretty simple. So anyway, okay, yeah, so you have a beautiful, complex but manageably complex system in your mouth, and yeah, I mean, the advice as somebody who has co-evolved with my microbiota for a long time, I would say, physically remove them. That's good, but maybe don't chemically attack them. So I'm a believer in flossing. I'm going to sound like a dentist. You should floss your teeth.
Oh, yeah.
Because you want to keep them away from your gums. You want to keep the bacteria away from your gums so that your gums don't react. So floss, yes. Tongue cleaning seems to be a good idea too. Actually, it seems to shift the microbiota in the direction of being better at manipulating nitrate. We think that we have to do more research on that. But yeah, so physically getting rid of them, that's really good. Yeah, floss your teeth.
I think that's wonderful advice. I won't make any comments about people that are using white strips and the effect on the oral microbiome of that. Probably is. But it is certainly a newfound respect that one can have when they blearily look at themselves in the mirror as they brush their teeth. And I have had the most wonderful time with you here today. I want to thank you for the beautiful images, your clear explanations, and the relevance of what we discuss to people who aren't trained as microbiologists, because I want to emphasize it's easy to be like that character in the movie The Right Stuff, where one of the astronaut trainees says, "What are you doing?" And the guy says, "It's complicated." No, it's wonderful. And the enthusiasm that you show is, if you'll forgive me, infectious.
Well, it's been a really, really great pleasure talking to you. I really enjoyed it. Thank you so much.
Thank you so much again. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always with epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Jessica Mark Welch can be found at the ADA Forsyth Institute in Somerville, Massachusetts. Many, many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.